right into this explainer. Today, we're going to demystify an absolute high-stakes logistical nightmare that is happening inside you right this very second. We are talking about cell division. It's literally happening millions of times a second, and yet the precision required is nothing short of miraculous. We're talking about duplicating and perfectly separating an entire genome, and the physics and chemistry behind it are absolutely fascinating. We're breaking this massive operation down into 5 manageable phases: the ultimate balancing act, mechanics of the spindle, the quality control checkpoint, waiting for the go signal, and finally the final split, also known as cytokinesis.
So let's kick things off with phase 1, the ultimate balancing act.
To really get this, we have to realize that splitting a cell isn't just biology. It's a literal extreme physics problem. It's like a microscopic construction site where the scaffolding has to be incredibly strong, but also super dynamic. It begs the question, how on earth do these microscopic cells generate the immense physical force needed to perfectly pull their DNA apart without snapping it? I mean, proportional to their size, the forces we're talking about are basically like moving mountains. Well, it turns out the secret lies in specialized motor proteins called kinesin and dynein. Picture these as tiny, microscopic engines that literally walk along cellular cables known as microtubules. They are the actual muscle of the whole operation, generating all the push and pull forces needed to maneuver those chromosomes into perfectly aligned positions.
Moving right along to phase 2, mechanics of the spindle.
Let's zoom in a bit on the actual mechanics of this structure, the mitotic spindle. It's an absolute marvel of temporary cellular engineering. What's wild is that it's basically a microscopic tug of war. You've got kinesin motors pushing the microtubules apart to create an outward force, right? But at the exact same time, dynein motors are pulling the anchor points, the centrosomes, together, creating an inward force. Throw in the polar rejection forces from growing microtubules literally pushing the chromosomes, plus the attractive tension from the kinetochores pulling them in, and wow, you end up with this incredibly delicate, highly regulated mechanical balance of 4 distinct forces acting all at once.
Because of all these opposing forces, researchers modeling this system found something pretty incredible, a regime called bistability. Basically, think of it like a highly advanced shock absorber. The spindle's tension network is so versatile, it can actually stabilize at 2 completely different lengths depending on the conditions it faces. So it's not just some rigid, static structure. It's a flexible, adaptable machine that finds its mechanical stability based on the delicate balance of those inward and outward sliding forces.
All right, phase 3, the quality control checkpoint.
Okay, so the physical structure is built, the cables are attached, the tension is dialed in, but who actually gives the order to start pulling the DNA apart? Because a premature pull, that would be absolutely disastrous, leading to unequal genetic distribution. As the research literature emphasizes, the physical connection between the microtubule cables and the chromosomes at a site called the kinetochore is, quote, joined at the hip with the cell's communication system. The physical attachment site is quite literally the biochemical signaling hub. And that brings us to the spindle assembly checkpoint, or the SAC. You can think of the SAC as the world's strictest, most uncompromising microscopic bouncer at a club, or maybe a hypervigilant quality control inspector on a factory floor. Its one and only job is to ensure every single microtubule cable is fully, securely attached to every single chromosome before allowing cell division to happen. If even one connection is missing or faulty, boom, the SAC shuts the whole party down and completely halts the operation.
Let's trace how this chemical cascade actually builds. Now, you don't need to memorize the chemical names to appreciate how elegant this is. Listen to this. Just one unattached kinetochore is enough to trigger a massive cellular alarm. It grabs a protein called MAD1, which acts like a catalyst to rapidly convert other free-floating proteins into a super-active state. All of these combine to form a massive supercomplex known as the MCC. And the main takeaway here? This MCC acts as a direct, incredibly powerful stop sign. As long as it's being produced, the cellular machinery needed to split the cell is completely frozen in its tracks.
Which leads us to phase 4, waiting for the go signal.
So the cell is perfectly poised. Tension is high, but it has to wait patiently for that absolute all clear. It is a high-stakes waiting game. The crucial transition goes something like this. At first, those unattached chromosomes act like little factories, just pumping out that MCC stop signal. But as time goes on, and more chromosomes manage to attach properly to the microtubes, the number of those signal factories drops. The chemical stop signal slowly diminishes, until finally, every single chromosome is perfectly attached. The MCC production stops completely, and the cell gets a full green light to proceed into anaphase, which is the actual physical separation of the chromosomes.
You can kind of conceptualize this whole process as a rapidly dropping chart. In early mitosis, those MCC chemical levels are sky high. It's a hard stop. By mid-mitosis, as more connections form, the levels naturally start dropping. And the exact moment that final chromosome is securely tethered, MCC production halts entirely. The levels plummet effectively to zero, and that glorious go signal is finally realized. And finally, phase 5, the final split, cytokinesis. Once that go signal is achieved and the chromosomes are successfully pulled to opposite sides, the cell faces its very last physical hurdle. I mean, it has to actually become two completely separate entities, right? This brings us to cytokinesis. If mitosis was the careful duplication and separation of the master instruction manuals, cytokinesis is the dramatic grand finale where the cell's main body, the cytoplasm, is physically cleaved in two to build the two new houses. It is honestly amazing how different life forms go about solving this final physical problem. Animal cells, because they have flexible membranes, literally pinch themselves in half using a contractile ring made of actin. It creates a cleavage furrow, kind of like pulling a drawstring bag incredibly tight. Plant cells, on the other hand, are surrounded by a rigid cell wall. They can't just pinch, so they have to construct an entirely new brick wall, a cell plate, right down the middle to separate the two new daughter cells. But despite these wildly different engineering solutions, pinching versus building a wall, both plant and animal cells share fundamental underlying similarities. Both of these processes occur right after mitosis. Both guarantee the genetic material we just so carefully separated is safely compartmentalized, and both rely heavily on the exact same underlying microscopic architecture of the cytoskeleton. Specifically, they're both utilizing those microtubules and actin filaments to get the job done. And that really brings us to the ultimate why this matters. If we can fully map out and understand this incredible logistical nightmare, the precise physics of the tug-of-war, the intricate biochemistry of the spindle assembly checkpoint, the mechanical transition of cytokinesis, could we eventually hijack these very systems? Could we find new, highly targeted ways to permanently flip that stop signal and halt the rampant, unchecked division of cancer cells? It's a deeply provocative thought, and it's exactly why digging into these fundamental biological mechanisms is so absolutely crucial. Thanks for joining me for this explainer, and I'll catch you in the next one.